Okay, so carrying on, there's a method to visualize these chromosomes via G-banding. So we typically use lymphocytes to get a hold of chromosomes. So firstly we have to extract the lymphocytes. We culture at 37 degrees Celsius for 3 days. We add clotrimazine, why are these words so hard? Clotrimazine to arrest the cells in metaphase. We add hypotonic saline to cause cell swelling, the cell lyse, and we mount onto a slide and we stain. Simple. I'm now going to go on to a condition in which G-banding can tell us about, which is called Philadelphia chromosome. So basically, Philadelphia chromosome is where, as we can see on this example, here are two chromosomes, chromosome number 9 and chromosome number 22. Chromosome number 9 has a gene called ABL, chromosome 22 has a gene called BCR. So what happens is the ABL gene is on the bottom of chromosome 9. It's a proto-oncogene, meaning it's a normal gene that can become an oncogene. So if there is translocation between the chromosome 9 and 22, it causes the ABL gene to become oncogenic. Clear? And that's just an example of basically how we can use G-banding to show that, and that's basically here we can show that the translocation has occurred between those two chromosomes, chromosome number 9 and chromosome 22. As we'll be able to see that there's a different in size, there's a different in the banding patterns, except a different in size and different in banding patterns, as we talked about earlier. So, coming to a summary of G-banding, we use it to see how the karyotype, which is where the chromosomes are lined up from 1 to 23, and it shows us the number and appearance of chromosomes. So we can use it to see how the karyotype of the patient differs from the norm. We can use it to look at aneuploids, which is basically a posh word for having a different number of chromosomes to the normal 46. We can also use it to look for translocations and very large deletions. So G-banding identifies big abnormalities. That's the most important thing. G-banding identifies big abnormalities. If I can just get that to focus, big abnormalities. We then move on to the next type of investigation, which is called FISH. FISH stands for fluorescent in situ hybridization. So similarly with G-banding, it also uses cultured cells and metaphase chromosomes. However, it can detect changes that are microscopic, 5 to 10 megabases, I believe. Yeah, just like megabytes, megabases, so 5 to 10 megabases, which is pretty big, but smaller than G-banding. So instead of using a stain, we use fluorescent probes to bind to DNA. So the most important thing basically about FISH is that it can detect changes that are microscopic. So a key point of FISH method is that we need to know what genetic deletion causes a disease in order to be able to test for it by standard FISH. And this is because we have to design a specific probe that will bind to the desired section of DNA. So those are just two main points. Basically, key point of FISH is that we need to know what genetic deletion causes the disease in order to be able to test for it. And because we have to design a specific probe that will bind to the design section of DNA. Clear? Okay, so the method of FISH is very similar to the method of G-banding. Firstly, we extract the lymphocytes. We culture at 37 degrees for 3 days. We add colchicine to arrest cells in metaphase. We add hypotonic saline to cause cell swelling. The cell lyse we mount on a slide. We then denature the DNA so they become single-stranded DNA, and we add probes. So we denature the DNA and we denature the probe to ensure both are single-stranded DNA, or both are single-stranded, so they will bind and anneal with each other. This will produce a fluorescent marker in the part of the chromosome where the probe is complementary. So the probe will bind to the part of the chromosome which is complementary, and it will cause it to flash up. So FISH is good for identifying microdeletions, for example 22q11 del syndrome. Prader-Willi syndrome on chromosome 15, and Kreidukrat on chromosome 5. So I'm going to show you basically how fish actually works in real life. This is how Kreidukrat would be identified, and as you can see on the left is the G banding, and we can see anything that would go wrong. On the right is fish, and it will show you the actual where the actual genes are, and it will show you basically that on the left hand side you can clearly see that there the genes are present as a probe has binded and annealed to the complementary DNA, whereas on the right it's clear that the genes aren't present. So this is how clear that fish is. So we'll move on onto something called spectral karyotyping, aka SKY. So this allows all chromosomes to be looked at once. This means a translocation disorder is easy to spot. I'm just going to show you what SKY looks like. If I can get a quick picture. Where is SKY on this? Let me see. So let me go through the. So there's Kreidukrat, and here is spectral karyotyping, aka SKY. So as we're saying, this allows all chromosomes to be looked at once, and it means that a translocation disorder can be easy to spot. And how translocation disorder can be easy to spot is that if each one has a different colour, then we'll be able to see basically that the mixed match of colours. So there, if you see number 9 and number 10, that might be a translocation because it looks like the different chromosomes have different colours. I'm sorry, but the picture is very bad. But anyway, to summarise fish, so we use fluorescent probes for specific parts of the genome. We're looking for abnormalities like aneuploids, translocations, and large deletions. So as we said, aneuploids are having a different number of chromosomes to the normal 26. So we use fish to identify aneuploids, translocations, and large deletions within a chromosome. So large deletions within that specific chromosome. And finally, with traditional fish, we must know what is normal and what is the cause of the disease, what the cause of the disease is. So we designed a probe to fit the normal gene. So that's maybe a negative of fish, is that you have to know basically what you're looking for. We have to know what the cause of the disease is. We have to know what the cause of the disease is so we can design a probe to fit the normal gene. Capiche? Okay, great. So we move on to QFPCR. So QFPCR stands for Quantitative Fluorescent PCR. It's a quick way to determine trisomies using microsatellites. So what are microsatellites? Microsatellites are short repeated DNA sequences which appear a lot in the genome. Microsatellites are short repeated DNA sequences which appear a lot in the genome. So in large areas of the genome we have repeats which can be di, tri, or penta, hexa, etc. So we have these repeats which can be di, tri, penta, hexa, etc. So this would be called dinucleotide sequence, or trinucleotide sequence, or tetranucleotide sequence, or pentanucleotide sequence, etc. So here is basically a microsatellite, an example of a microsatellite. So we have two unique flanking sequences on either side of the microsatellite. We have the microsatellite in the center, which can be di, for example, can be CA with a specific number of repeats, which is called the variable number, which is shown as n. Or it could be tri, such as CAT, or tetra, like CATG, something like that. And it can be hexa. So the number of repeats is constant within an individual. So the number of repeats of the microsatellite itself is constant with an individual. So for example, in myself, I might have the repeat of microsatellite 10 times, whereas in someone else, it might be 11 times. And the number of repeats vary between individuals. Just as I said, it might be 10 times in myself, and it might be 11 times in someone else. We're typically heterozygous for these repeats. For example, the 10 repeats may come from dad, and 20 repeats may come from mum. microsatellites always have unique flanking sequences, as we showed you before, and unique flanking sequences are completely unique to the microsatellites, allowing us to recognize the microsatellites on any particular chromosome, and whether it's from the mother or from the father. So unique flanking sequences are completely unique to the microsatellite and allows us to recognize the microsatellite on any particular chromosome, and whether it's from the mother or from the father. So with a trisomy, so we'll go on to an example of how we can use microsatellites in QFPCR, or quantitative fluorescence PCR, so many complicated words. So, for example, with a trisomy, we will see 3 copies of the microsatellites on 3 different chromosomes. So here's an example of a mother, a father, and the 2 children. So the mother has microsatellites, one of 12 repeats and another one of 9 repeats. The father, one of 7 repeats, one of 11 repeats. The 2 kids, as you can see, one has 2 chromosomes and one has 3 chromosomes. You can clearly see there's trisomy, and we can detect 3 copies using QFPCR. We tag the microsatellites with fluorescent tags. We use machinery which can detect how many peaks, which indicates how many copies of chromosomes there are. So we can use machinery which determine how many peaks, which detect how many copies of chromosomes there are. So 3 peaks would be trisomy. So I'm going to show you a quick picture of what it would look like. And here we can see clearly. So as I was saying, normal, there would be 2 peaks. As we run these, this QFPCR through the machinery, we'll be able to see how many peaks. The 2 peaks would be normal, the 3 peaks would indicate trisomy. Clear? Okay. We then move on to the summary of QFPCR. So basically, QFPCR, it tells us how many copies of chromosomes the patient has. So we use fluorescent probes for specific microsatellite markers on specific chromosomes. We use fluorescent probes on specific microsatellite markers on specific chromosomes. It's quick, it utilizes extracted DNA, and however, you need to know what you are looking for so you can create the probe for it. We then go on to the final investigation for, I can't remember what it was called. It was called cytogenetics. So we'll go on to the final investigation for cytogenetics. It's called Array CGH, or ACGH. Array CGH stands for Array Comparative Genomic Hybridization. Wow. Array Comparative Genomic Hybridization looks for microscopic chromosome abnormalities such as microdeletions, microduplications, and copy number variations. So Array CGH, it mainly identifies developmental problems and dysmorphia, which is a deformed body shape. Cool. So notably, something important, CNVs, or copy number variants, tend to be responsible for this. So copy number variants tend to be responsible for developmental delays and dysmorphia. Copy number variants are a form of microdeletion and microduplication, as there may be an increase or decrease in the number of bases. So copy number variants are 10 kilobases to 5,000 kilobases. Copy number variants can be protective, e.g. from HIV, or detrimental, e.g. may lead to the cause of autism or schizophrenia. Many CMVs do nothing and are present in 12% of the genome. So CMVs make up 12% of the genome, so most of them do nothing. Okay, so the simplest CMV is the presence or absence of a gene. An individual genome could therefore contain 2, 1, or 0 copies. So here's our first example. Here's our copy number variants. Here, this one has 2, this one has 1, this one has 0. So for the first example, the copy number variant for this gene, for example, maybe CFTR, would be 2. So the normal copy number for a gene would be 2. 1 gene from the mum and 1 gene from the dad. And each gene would be on either allele. But if one gets deleted, so if one gets deleted, the copy number will become 1, and if both get deleted, the copy number will become 0, and that can cause the potential autism and schizophrenia. We'll go on to the second example, which may be an alternative in what occurs is that you could have duplication of genomic segments, which could result in a diploid copy number of 2, 3, or 4. So we'll talk briefly about the process of array CGH. That's wrong. It should be CGH. Array comparative genomic hybridization. So number 1, the patient and the control DNA are labeled with different fluorescent dyes and applied to the microassay. So as we can see, patient DNA has a green fluorescent dye, the control DNA has a red fluorescent dye. The patient and the control single-stranded DNA must denature single-stranded DNA. It's single-stranded DNA because it must because the DNA has to basically denature. So the patient and the control single-stranded DNA will compete to anneal with the probes on the microassay slide. So basically we have these two DNA, we add them together and we add them to the microassay slide to be able to hybridize. So the patient and the control single-stranded DNA will compete to anneal with the probes on the microassay slide. What should happen is there should be an equal hybridization or binding to the slide probes, and there should be 2 copies of each DNA of each gene, and 2 copies from the patient and 2 copies from the control. Therefore, the relative fluorescence should come out as being equal, as affinity of both sets of DNA should be the same, if they both have 2 copies of each gene. You probably heard that and you're like, what the hell? It's so simple. So we have 23 pairs of chromosomes in this DNA. 23 pairs of chromosomes in this DNA. So we have basically, they're able to all hybridize to basically complementary DNA here. Each, if I'm getting this right, so the probability that each should be able to compete with each other should be equal. In normal, for example, if I had the gene for brown eyes, so the patient has DNA for brown eyes, the control has brown eyes or whatever, then there should be 2 genes for brown eyes, 2 genes for brown eyes here and here. So both of them would compete equally, and we would get an equal hybridization because they both have the genes for brown eyes. However, if there is, what does that say? If there is deviation, I'm so sorry, that is terrible handwriting. If there is deviation, and the fluorescence of the microassay is different, it indicates there is a duplication or a deletion, causing the change in fluorescence. So, for example, if there may be DNA loss or DNA gain. For example, if the DNA patient somehow only has 1 gene for brown eyes, therefore it would indicate that there would be DNA loss, and that more of the genes for brown eyes from the control DNA would compete and bind to the probe. On the other hand, if the patient DNA had more genes for brown eyes, it would bind more, and it would indicate DNA gain, basically. I hope that makes sense. If it doesn't, then read over it again, ask whoever's next to you, and inshallah should be fine. Yeah, and we'll move on to the final part of the lecture, which is basically looking at, as we said, the molecular genetics. There's 2 types of investigations for molecular genetics: Sanger sequencing and next generation sequencing. So Sanger sequencing. Okay, my side note says take them through the slides. I'll try and take you through it as quick as possible. So Sanger sequencing. So it's targeting and sequencing an individual's gene. That's what Sanger sequencing does. What's needed is the patient's DNA, which is the DNA template, DNA primers, the DNA polymerase, also known as TAC, DNTPs, which are deoxynucleotides, DDNTPs, which are dideoxynucleotides, which have no hydroxyl groups, which means that extra oxygen is removed, so there's no elongation of the chain, as it cannot form a phosphodiester bond, and termination of the chain occurs. So before anything, we need to isolate the DNA and amplify the genomic bits we're interested in. The DNA should be free of protein and cellular debris. Amplification is via PCR or cloning, so the amplification of the DNA is via PCR or cloning, and the Sanger sequencing is made up of 4 parts. So the first part is strand separation. Wow, so the first part is strand separation. We have double-stranded DNA and we need to separate it. So we have double-stranded DNA and we need to separate it. So there' our complementary strand and there's our template strand. This important point that you'll need to know for everything is that the template strand will normally be going 3' to 5' and the complementary strand will be going 5' to 3'. And the reason for that is when we add the primer is going to basically join. The primer basically needs to start 5' because when you do polymerization it goes 5' to 3'. So imagine we have the primer added here and the primer will be going 5' to 3' that way. But we're going to talk about that in the next section now, where we anneal the primer. So our primer has to be complementary to the 3' end as DNA polymerase act 5' to 3' direction. So as I was just saying, we have to anneal the primer onto the 3' end of the template DNA as the DNA polymerase will move in a 5' to 3' direction. So this is 5' for the primer and this is 3', so it will move that way, DNA polymerase. Yeah, exactly, there we are. So number 3, there will be extension. So we add our DNTPs and our DDNTPs and our Taq polymerase. So the DDNTPs, excuse me, are fluorescently tagged. So the Taq polymerase will cause complementary strands to build up via DNTPs. However, eventually a DDNTP will be added and extension will stop as there's no OH group on the DDNTPs, as we said before. So basically here the black ones are DNTPs, the ones with the stars are DDNTPs. They will join. Where's the picture? They will they'll start joining and extending the chain until a DDNTP is added. Cool. And then there will be termination. So number 4, termination. So however, what will happen is that we have millions of template strands due to PCR, so they will not all terminate in the same place in the chain. So basically, as this DDNTP binds, this chain will terminate. However, there's so many chains that not all of them will terminate in the same place, and not all of them will terminate at the same time, and that's what you get basically. So this one has terminated here, this one has terminated here, this one terminated here, and here, and here, and you've got so many you've got so many DNA templates that you'll basically have a profile where you have the whole sequence. So you'll have this coded, it's so hard to explain, but basically you'll have your DDNTPs which will attach to each section of the template. And what will happen is that, if I can show you, so after that basically is that the individual strand will be terminated. So if you just go back a section, because I'm jumping ahead, the individual strands is terminated, but other strands by chance will go longer, will go on for longer, or will even go on for less. So we end up with fragments of every possible length of the exon. Cool. And what we do now is that we now have double-stranded DNA of every possible length, but we only need the strands with the fluorescent tags. So we denature the double-stranded DNA as seen. So here are our fluorescent tags, here's our double-stranded DNA. We want to denature it as we only need the strands with the fluorescent tags. So we denature this double-stranded DNA and we keep the part which has the fluorescent tag attached. And as I was saying, you do this and basically you'll have every possible length of the exon with a DDNTP attached at the end. So you have every possible length of